las 5:30, 6 menos cuarto, como muy tarde, como hemos anunciado. Por favor, doctora Elkin.
Bueno, buenas tardes. Espero que hayan tenido un buen almuerzo. Y bueno, después del almuerzo siempre es un poquito pesado, así que traten de moverse un poquito para despertarse. Nuestro siguiente presentador es Gino Gramp, que es miembro del IFMA, director de políticas de investigación y desarrollo sobre productos biosimilares de Amgen, y él va a tratar el tema de medicamentos bioterapéuticos y farmacovigilancia. Bienvenido.
Muy buenas tardes. Lo siento que seguimos otra vez con farmacovigilancia después del almuerzo, pero trato de ponerle muy interesante. Pero lo siento que no soy tan buen mozo que el Tomás de Roche le doy la exposición en inglés porque no tengo, me falta el vocabulario técnico en español. Bueno, ahora. So, switching to English. The themes today are again on pharmacovigilance. And first I will start a little bit talking about the scene. I think we heard also earlier this morning about the pharmacovigilance, the status of pharmacovigilance around the world and how that relates to biologics regulations. Then I'll specifically focus on the challenges for biotherapeutics, for having multiple source biotherapeutics in a country, and it can't be, pharmacovigilance can't be treated the same way as it is maybe for generic drugs in that situation. And finally, focus on the thoughts from IFPMA about identification and traceability. So, obviously, as we heard, there's pharmacovigilance systems being established around the world, some places more advanced than others, but it's good progress to see the progress from UMC in many regions, including Latin America. So this is good, but some countries still need to improve the rate of reporting and the accuracy of reporting, I think, in order for this to be effective. Another thing that happens at the same time, especially for biotherapeutics, is that the system of INN, traditionally, for complex glycosylated products, the type of product we heard about from Dr. Murillo, the INN system had a rule that if you had differences in glycosylation, you'd receive a different Greek letter, como alpha, beta, gamma, see? But this system isn't sustainable and it's not being enforced, so all these non-comparable products, even though they're not comparable for glycosylation, they still receive the same INN in all around the world. So that system, the traditional rules from WHO are not being followed. So this is a challenge. And we need to have robust pharmacovigilance. So how do we establish robust pharmacovigilance? Well, certainly the countries need to have guidance, right, to ensure patient safety, guidance that manufacturers follow, and also guidance for how pharmacists and medical doctors should report into the system. We need to improve the identification and the naming of these products. We just heard about some of the confusion that could occur if the identification and naming is not clear. And of course, the most important part, I think much more important even than identification, is to have better collection of the data, right? To emphasize collection of the data, reporting, surveillance, signal detection. Because if nobody's looking at the data, it doesn't do any good. So, and finally, to focus on the risk and the benefit of doing this. So, before I talk about the challenges of biotherapeutics and why they need a special attention, I did want to mention this isn't just IFPMA's idea that this is important. I think it's obvious when we hear all of the lectures from yesterday and today why it might be important to have extra pharmacovigilance for biotherapeutics. But this is also what the European Commission has said in their pharmacovigilance legislation from 2011. They said that you need to have product-specific tracing so that each product has its own signals for biotherapeutics. And the FDA, in thinking about biosimilars, which are coming also to the United States, said a similar thing. So this is governments as well as manufacturers and IFPMA believe this is important. I won't go over this slide in detail. We heard yesterday many examples of the differences, of course, between biotherapeutics and chemical medicines. But I will emphasize two important themes. We heard about complexity, right? Yes, it's very complex. What does that mean? You may not be able to detect every difference or understand every difference between two biotherapeutics before they're approved. But also, and this is my background is in manufacturing, sensitivity. These products are very sensitive to their manufacturing process. I have spent many, many years looking at manufacturing processes, things going wrong in manufacturing processes, trying to fix them, and the products will have quality issues that you have to investigate. Those things, most of the time, have no safety issues, right? Otherwise, we'd have a big problem. It's very rare, but every once in a while, it's true, we can have a safety problem from associated with a manufacturing issue. And they're very sensitive, biologics. Even the change in, we heard from a stopper in the syringe, or the change in the amount of tungsten that could be in the glass syringe barrel could affect the safety of the product. So because of that, I think that's one of the biggest challenges of biotherapeutics. They're sensitive, they could change over time. We need to monitor them. Immunogenicity. We heard from Barry Cherney yesterday a lot about this, so I won't go into detail. Just to remind everyone that immunogenicity can vary tremendously from one product to another, one product class to another. For some products, it's very rare that any patient would have antibodies. For other products, it's very common, and it may not even harm the patient. It could just be part of the natural therapy that they have anticuerpos, no? Right? But sometimes immunogenicity can affect the patients. Immediate reactions that could be very difficult, or longer term, they could lose the efficacy of the product, right? So these are important things, and they may not be the same between a non-comparable product or biosimilar product and the reference product. We need data, but also they could change over time, perhaps. So again, pharmacovigilance could help. Complex pharmacology. This is mainly an issue for the monoclonal antibodies, because think about erythropoietins, filgrastins. They have very simple pharmacology. They bind to a receptor, you get red blood cells or neutrophils. That's not what I'm talking about. For the monoclonal antibodies, Professor Murillo went over this yesterday. They can have many functions. They can have important functions not only on the FAB, where they bind the target, but also on the constant domain at the bottom of this figure, right? They can bind to parts of the immune system that can affect the way that medicine behaves in the body. It's very hard to understand all of these effects in vitro. We have very good assays. We're developing better science, but still to predict how those things translate at the individual patient level. In fact, the publication here I cited by Giesen in 2008, they looked at long-term safety issues that emerge for biologics after approval. And in many cases, the kind of the unexpected or emergent safety issues relate to immunomodulatory effects. Either the medicine increases the immune system activity in certain patients and causes a difficulty, or the medicine suppresses the immune system in certain patients and causes infections or something. So these complex pharmacology could be important. Probably the most important challenge for biotherapeutics, and we heard, again, many talks about this, is that the analytical and the preclinical studies, right, they're very good. They're very important for the comparability exercise. But at the end of the day, we still, with current science, we can't predict everything about whether a difference that we observe could be important in patients, especially for immunogenicity, right? But also even pharmacology. there's many examples, unfortunately, many examples of companies making a change to their manufacturing process and finding out that the bioavailability, the pharmacokinetics changed. So, this can happen. So we're still learning. We can't predict this in animals very well. Okay. And the final challenge, there's 5 or 6, I can't remember, is, of course, the different regulatory pathways we've been hearing about for 2 days now. If you're tracing these products, do you put them all in one pool and call them all biosimilars and originators with the same INN? Because that's really the state of the art, I think, is in some cases, is to aggregate them all into one bucket. Rituximab, epoetin, and look for safety signals. And that's okay. We've always done that for medicines, yes? If you have Lipitor, atorvastatin for cholesterol, it's okay to aggregate the originator and all of generic product signals to see if there's a toxicity or a drug interaction or something. It's fine. That's fine. There's nothing wrong with it. And it seems true for biologics. But we also have to disaggregate the signals because they could be different. We have originator products, and sometimes more than one originator product, and nobody ever showed that they were similar to each other, but they could have the same INN, like interferon beta, interferon alpha. There's multiple products, originator products with the same INN. Of course, biosimilars. At least they showed that they were similar to one reference product, right? But they never showed they were similar to each other. So it might be important to track those on an individual basis, even though they're assumed to be safe and effective. And of course, the non-comparable products that are quite common in Latin America and other regions. Sometimes these products are approved with some data, some good data, but they've not shown to be similar, highly similar to another product. So it should be traced at the individual product level. I won't go over this slide. We all know, I think, what a biosimilar is now. After 2 days, we'll have a quiz after this, an examen. So, what are the implications for pharmacovigilance, right? Well, many countries are trying to align with WHO guidelines for biosimilars, but not everybody has figured that out yet. We're still developing, right? And sometimes we already have legacy products, so we already discussed that. Legacy products that did not meet the biosimilar pathway, right? So what happens is multiple source biologics, many biologics sharing the same INN in the same country. And if they're being tracked at the brand name, it should be fine, right? If everybody's careful to prescribe by brand name, dispense at the pharmacy by the brand name, the patient knows what the brand name is, the prescriber knows, then the pharmacovigilance maybe can work. But if people start going to generic-like thinking and write the INN because it's easier, because there's less problems with the pharmacy, who knows? It could cause a problem, right? Also, sometimes these products are not highly similar because if they've not followed the similarity pathway, they could use different doses. They could be approved for different indications. We heard about in Brazil, right, that one of these products, the individual pathway product, will only be approved in one indication, right? It's not going to be exactly all the same indications as the originator product, and different safety profiles. So it's very important to have accurate identification of these products, hopefully the brand name, but maybe other identifiers too. Good record keeping, both in the pharmacy and in the prescriber's records. And of course, education, physician and patient awareness that their safety is not just a privilege, it's a right? So it's not just a privilege that you can get these advanced medications. It's also a right that they should be good quality. And then if there's a problem, that you should feel confident to report the problem, right, to the company so they can investigate. So, as I mentioned earlier, it's also important for risk management because biologics, biotherapeutics have more risks possibly in the manufacturing than a generic pill. They're so complex, they're sensitive, they're injectables, many factors that make them risky. So I think I went over this material already below. But originator products, similar products, and non-comparables, they each might have a different amount of data that need to be evaluated after approval to see if there's a difference that wasn't detected. One of the problems, I think, for some of these medicines, think about interferons for multiple sclerosis or the anti-TNF for rheumatoid arthritis. These products are chronic. The patients need the medicine for years to maintain their condition stable. Now, if you have a lot of competition, which is good to reduce prices, improve access, right? What can happen is the pharmacy can switch, or the prescriber can switch the patient from one medicine to another over a 1-year period. If there's a long-term immunogenicity issue, which I believe somebody asked just before the break, that this could take a year sometimes to the immune system to mount its maximum response. And so if the patient has been on 3 different medicines in the previous year, it could make it more difficult for the pharmacovigilance system to identify which medicine may have had a new signal, a higher signal of immunogenicity. I'm not to say that it's not appropriate to switch the patients, but just as something to keep in mind in the system to have good record keeping. I've cited a paper here that was interesting recently about a patient who had an immune response in Europe, and they put the patient on 3 or 4 different biologics for erythropoietin. By the time they realized the patient was getting worse and worse, their hemoglobin was going lower and lower and lower. By the time they diagnosed the problem, the patient was already on the 3rd drug, but because they did keep good records, they were able to go back to and tie it probably to the first medicine. I want to give an example this is a real example from Latin America about interferon beta, and I'm using this to illustrate the challenges of the non-comparables, both from post-approval monitoring as well as how do we view these medicines. So these, as you may know, there are 2 originator products with interferon beta 1A. Again, they're not the same dose, they're not the same indications. They both treat multiple sclerosis, but there's some differences in the route of administration and the dose. In addition to that, there are several non-comparable intended copy interferon betas. And 2 years ago, some academic lab in Europe tested some of these products for purity and potency. So on the left of the figure is a purity, a graph showing the purity by SDS page. It's a gel chromatography. And some of the products have many more impurities than the originator product. Does it matter for the patient? I don't know. I'm not to say whether or not it matters for the patient, but it shows they were not comparable from a purity perspective. But probably more important or more significant, they also looked at the potency using a mouse potency assay. And some of these biosimilar, or I should say non-comparable products, had 1/3 to 1/3 the potency in the mouse assay compared to the originator product. These are products that are sold, these are on the market, including in Mexico and Colombia, and I go to my next slide to talk about that. So after that was published, I think one of the originator companies, certainly not my company, but one of the originator companies decided to go look at what's called observational study, because you can't even do this from pharmacovigilance. You can't look to see if the difference in purity and the difference in potency could cause a pharmacovigilance signal. It's very difficult to do this. So they did what's called observational study. They went to find the patients that were on the medicine and did some testing of those patients. So their idea was to find 90 patients each, 90 patients that had been on the originator product for at least a year, stable, with no switching, because otherwise the immunogenicity signal is completely mixed, right? And 90 patients on the non-comparable, on the copy product. They couldn't find them in Mexico and Colombia. They couldn't find 90. They found 30. Okay, so that's what they were able to study. They found the patients and they tested their blood samples for antibodies and also for some other markers to see if there were differences. So what did they find? The good news is, I guess the good news, they didn't find any evidence of elevated antibodies, but really the patient numbers were too small. Only 30 patients, probably not enough. You probably need 100 patients at least to detect that. But at least they didn't see anything alarming, right, for the copy product. But remember the potency data. The potency was 1/3. So there's a pharmacodynamic marker in these multiple sclerosis patients, and that's the figure on the right. The blue is the marker data for the copy product, and the red is for the originator product. The marker was lower, significantly lower, according to statistical testing. And what's interesting to me, I saw this is where you might see something in pharmacovigilance. Apparently the interferons cause maluca, flu-like symptoms, because that's the mechanism of interferons. They're not very pleasant, right, medicines to take. And the originator product, 80% of the patients reported that they had the flu-like symptoms, but the copy, only 30% had the flu-like symptoms, which means the product probably wasn't as potent. So this is difficult. This shows the difficulty of tracing these products after they're approved. It's very difficult to find patients only on one medicine, and even then the signals are difficult to interpret. So this is the challenge we all have. So in terms of traceability, flipping to the IFPMA's position on traceability is ideally we have prescription by brand, right, for biotherapeutics. Right? Hopefully that should be policy everywhere. Prescription by brand. Pharmacies trained to dispense the product by the brand. And if they have a less expensive product, call a physician and get the prescription changed. But don't just treat it like a generic, right, with substitution by INN. All right. If that happens, the records are clean for pharmacovigilance. The doctor knows, the patient knows, the pharmacist knows which medicine was dispensed and administered to the patient for when the adverse event report goes in. On the other hand, if you have prescription by INN, which happens, right? There's many countries it's either required, encouraged, tolerated, depending on the situation, right, to have prescription by INN. Some percentage of these medicines will be treated as a kind of a generic concept. Pharmacists give the patient whatever, right, as long as it's the same active ingredient. So in this situation, it becomes more difficult to trace unless you can go back to the pharmacy and identify exactly what medicine was dispensed. So record keeping is more difficult.
5 minutes, please.
Okay. Yep. So what does this have to do with naming, nomenclature? IFPMA supports the goal of, or at least one of the goals of the INN system. We support all the goals, but one of the specific goals of the INN system is to ensure that we identify the active ingredient. You could say, well, all the biotherapeutics have the same active ingredient. That's one way of viewing it, that the non-comparable products, the two originator products that were developed independently, and the biosimilar products all have the same active ingredient because they have the same amino acid sequence. I'm a scientist. I don't believe this for 1 minute, that they're the same active ingredient. They're similar active ingredients, but you really are not identifying the active ingredient. We could argue about that, but IFPMA believes that we should view biotherapeutics as having related but distinguishable active ingredients, and maybe the INN system can reflect that in some way, but not as a scientific judgment, as a traceability judgment. So if we can identify products both by multiple systems, not only the brand name, but also other things like the ATC code, batch numbers, or an INN, we can maybe improve the system. So to summarize, I think due to their unique characteristics, biotherapeutics, right, they need additional measures for pharmacovigilance, both to make sure that we don't have intrinsic differences in safety even when they're approved, because the amount of data may be limited when they're approved, but also that we can keep an eye out for changes in the safety, emerging safety issues because they're sensitive to manufacturing. Regulatory authorities can support effective pharmacovigilance through a number of ways. One, nomenclature. Either emphasize brand name, probably the best solution, brand name prescribing for biotherapeutics. Don't put them in the same bucket as generic drugs for these practices. Treat them with extra care. That's the best way. Or incorporating some identifier in the non-proprietary name if that's necessary. But important is the systems, to make sure the data are collected, that they're reported, that somebody is analyzing the data and looking at the data. It's not just going to Vigibase for UMC, right? As we heard, it's not their job to keep an eye on what's happening in one country with the products, right? It's each national authority that does this. And finally, advocacy, because this won't happen if patients and prescribers aren't educated, and pharmacists, right? Educated, informed, motivated to do more reporting for these products, because it's important to the system, it's important to safety for patients. Thank you very much.
Bueno, las preguntas las tenemos después de la siguiente presentación. Como sé que es un momento crítico ahora después del almuerzo, después de una presentación, les pido a todos que se pongan de pie, por favor. Y ahora tratamos de estirarnos un poquito para que la sangre fluya un poco más por nuestro cuerpo.
All right, I'll stand too.
Ya. Ahora, nuevamente, continuamos.
Was my talk that bad?
No. It's the lunch. Bueno, ahora continuamos con la doctora Rafaela Baloco. Ella es gerente del programa sobre denominación común internacional de la Organización Mundial de la Salud, y ella pues nos va a hablar de este tema que es la denominación común internacional, de la cual hemos hablado mucho en las últimas horas, bueno, desde ayer. Bienvenida.
Muchas gracias, señor presidente. Señoras y señores, me encantaría hablar con ustedes en castellano, pero mi lengua es muy limitada y no tengo el lenguaje técnico. Disculpa, mejor que le hable en inglés, aunque no sé mi lengua materna tampoco, pero por lo menos puedo decir la cosa con el justo término, con la justa palabra. Y como vamos a hablar de palabras, de nombres, sería mejor que pueda exprimirme lo mejor posible, sin hacer errores. I'm very glad to be here with you. It's my first time in Peru. I only was here for 2 days, but enough to fall in love with this country that I found very nice and with very nice people. And I have my watch, so I will try not to be said 5 more minutes and to keep the time. And if I am going too long, apologies. It's because I'm really passionate from my work, and I would like to transmit to you the passion and the fact that names are not only names, but they mean something in reality. And this is just a sentence that is spoken by themselves, by herself, or by itself, written by our Director General, that say that INN, it's an activity undertaken by WHO, and by doing this, we assure that WHO helps ensures that a prescription filled abroad is what doctor order back home. Inshallah, I would say, because after all what we have heard today, I'm not so sure anymore, but at least we try to do so. Just some basic facts for those of you who are not very familiar with WHO. We have 193 member states. Peru is one of them. We have 2 governing bodies, the World Health Assembly and the Executive Board, and the Secretariat, which is not only the one in Geneva, where I'm based, but we have 6 regional offices, 1 in Washington for OPS PAHO. And what we do, what the INN program does is based on the Article U of our Constitution. We have a constitution that says that WHO is responsible for setting standards for food, biologicals, and pharmaceuticals. The program of the INN is very old. It has the aim or the scope to provide one single name worldwide for active pharmaceutical substances. It was initiated in 1950, so it's full of inconsistencies because it's an old program. And it's operational since 1953. It is, as I said, based on the Constitution. And of course, we started with the role of harmonizing the existing nomenclature bodies that are more and more not existing, with few exceptions, or let's say, not that they are not existing, but they are relying on INN. And we started, if you would see the first list, they are very rudimental, just to say that this can be called like that, and not even the IUPAC or whatever, just the name. And now it's a bit more complex what we do. And a number of years ago, we named the first insulin human. So it's not yesterday that we started naming biologicals, it's quite some time ago. And this is just a repetition, but just to say that indeed the INN is a unique generic name, or should be a unique generic name, that it is recognized globally, and it is placed by WHO in the public domain. The INNs do not belong to WHO and do not belong to the applicants, even to the manufacturer developing them. They belong to everybody. And this is their strength, the fact that they are used in almost all member states and accepted globally. Who are we? Well, we are not alone, first of all, because we are just 7 humans based in Geneva, and we couldn't do that big amount of job or build this consensus if we wouldn't work very closely with many other stakeholders. Well, I will not mention because lack of time, but just, well, in addition to the regulators and the industry, I would like to make you an example, the fact that we collaborate with the World Customs Organization. Funnily enough, the World Customs Organization based their fee classification for medicines on INN. Certainly, this was not the scope when the INN program was established, but it is a factor. And of course, WIPO, the National Trademark Departments, and many, many other stakeholders. It's really an interesting job because I collaborate with so many people, and I have the chance to discuss and work with many different people from many different backgrounds. And what's in the name? Well, I will not comment this because in my opinion, a name is just a name, but there is a lot in a name. And certainly, a name that identifies a pharmaceutical substance includes all what is in the range of the pharmaceutical field, safety, rational use, etc., etc., etc. An INN is a unique name. Then it should be distinctive in sound and spelling from all other names in common use. It should not be liable then to confusion. It is formally placed by WHO in the public domain, and this is important. And I want to raise this. Maybe this is not really related to biologicals, but also no names should be derived from INN. No names, trade names, brand names, should include the INN stem, because this would infringe the program, the system. I saw that there was a rituximab, and I saw one of the brand name completely copied from the INN. Well, this shouldn't be. And we do what we can to try to protect the INN, not because we are jealous, but because we want this to remain a property of everybody. And of course, they can be used without any restriction to identify pharmaceutical substances. I don't know how many pharmacologists, I am one, how many pharmacists, how many medical doctors are here with us today, but I hope that you all studied using INNs or DCI, Denominación Química Común Internacionales, in not brand names. At least I did, and I trust you all did as well. The INN system is composed of a secretariat, of a group of experts. I think it's the largest nomenclature group of experts. There are 40 experts from all over the world, and with different background and expertise. And a number of years ago, because we were thinking that it was more and more challenging to name biologicals, we established an advisory group on biologicals to tell the INN expert how to name biologicals and how to define the policy for biologicals. And then we have the publications, which is our tool to disseminate the information. It's an old tool. We send a circular letter to all member states. This is sent by the Director General through us and the expert tool. And maybe you receive this list from time to time and you have the comment, yes, we agree, not, we don't agree. I receive replies from all over the world, so I guess you received the list. And of course, because we try to be a bit modern, we publish this on the web. We have our extranet. We have a lot of IT tools to disseminate the INN information. Why are we here today, or why am I here today? Because if you think that these are all data from 2005 and from the European Commission, but it's even more now. If you see the market or the medicines on the market, bio, there were in 2005 20%, but those under development were 50-50, and I think it's even more now. And look at our list. In 2006, we were naming 5, maybe a bit more percents of biologicals. But you see the trend in 2012, 2011, 40 to 45% of the INN that we assign are for biological medicinal substances. So this is an issue that affects us, and it's important. Not only the issue of biosimilars, but of biologicals in total. What is the current INN position for biological products? We have 20 biological groups with STEM. You have heard about rituximab, trastuzumab, I don't know how many MAB have been mentioned yesterday, and MAB is one of our STEM. But we have even 5 groups with nomenclature schemes, because the MAB, we are not happy to call them MAB. We then want to identify what is the target and what is the source. So we have 2 ZooMAB to say this is humanized, Zoo2 for tumor, MAB for monoclonal antibodies, etc., etc. And it's a bit more complex. And the fact that we have invented this fantastic stem of 3 syllables makes our life miserable when we have to invent a different name, because we have more than 300 MABs on the market, and the Latin alphabet has some limitations. And we have developed general policies for more than 10 biologicals groups. If you are interested, I hope you will be distributed the presentations, because in the line orange you see INN for biological and biotechnological substances review. There is an orange line, and if you will get it electronically and you will click on this, you will see all the, what we call a bio-review, so a collection of all our policies, of all our stems, of all what we have done in the biofield so far. And you can see that most of what is biological is included into the system, except the natural blood products, the immunoglobulins and the skin substitutes. And you see that it is at the end, and the traditional vaccines. Well, we have named some what are somehow called therapeutic vaccines, but the traditional vaccines are named by another committee in WHO, which is the Expert Committee on Biological Standardizations. And the cell therapy products, I think that this is something new I can communicate to you. We have decided that it is written under discussion, but I think I can share this with you. We have decided that we are going to name, we have almost agreed on a scheme. There are some raffination or some improvement that still needs to be done to try to harmonize as much as possible with the already existing scheme in the United States. And of course, don't read this, is horrible. But just to give you the flavor that all of what you use in your daily life is what we have named, because not only we have MAB, we have poietin for the erythropoietin type blood factors, we have a lot of the skin for interleukin type substances, and just to mention some of the most famous. These are some of the STEM is the syllables that identify the belonging to a certain group, either chemical or pharmacological. Chemical, it was in the past, now is more and more pharmacological. So these are the STEM that we have created for the biological substances. And we go now to the, I think, the hot part of the topic. What have we done so far for glycosylated proteins? We have decided, and I take the example of the poietin because it's a simple one, we have decided to identify, and so far it has always been like that, the group with the stem poietin. We have decided that if there was a difference in the amino acid sequence, we should have highlighted this in the name. That's why darbopoietin, because there was one amino acid difference. And alpha was, of course, to indicate glycosylation. And the glycosylation difference. So it was decided to indicate the glycosylation pattern with the Greek letter in full as a second word, alpha, beta, gamma, delta, and we have now until omega. Of course, I want to highlight this. The INN system is unfortunately a voluntary system. So all what is in the market, which is using an INN name, sometimes we have never seen, and perhaps we would have not name in this way. And I can just give you the flavor of one example, an epoetin alpha, which has been accepted to be alpha in the European market. They have some comparability guidelines, it's a biosimilar, etc. The same epoetin went into Australia, and with very similar comparability guidelines, similar to the Europeans, the Australians came to a totally different conclusion. And they said to the company, go back and apply for an INN. They didn't want, so as the result, TGA assigned a new Greek letter, lambda. And the same substance is on the market called alpha in Europe, lambda in Australia. Lambda is not an INN. Alpha, it is an INN. Perhaps it would have not been used by us if they would have come to us, because according to our policy for glycoproteins, that one has a different glycoprofile set by EMA, so we would have assigned a different Greek letter. And the same substance is called zeta, possibly the same substance in Japan, epoetin zeta biosimilar 1. So this is just one example, but that gave you the flavor of the confusion we are going to have soon. Well, of course, the Greek letters assigned in alphabetical sequence as they are received. We had in 2006 a meeting to discuss what do we do in the field of biosimilars, and we came to these conclusions that somehow are still applicable, which is the concept of biosimilarity is a regulatory concept and is not a nomenclature. I have heard a lot of discussions, but somehow I have to say it. It is depending on the decision of regulators. It is a regulatory decision, not a nomenclature. We name substances. We don't give any judgment to the substance. Generic substitution and reimbursement are still not WHO matter, are health policy matters, and depends on the regulators. Decisions on interchangeability as well, or substitution. Speaking the WHO language, interchangeability for me is when a physician does change the medicine. Substitution is when a pharmacist does. In some countries it's different, but it is what we call in WHO. So these are not done by WHO. INN are part of the pharmacovigilance system, not the only part. And there is not an INN policy for biosimilar. This is currently. However, we said that glycosylated proteins from different sources expected to different in their glycoprofile are given different name, and this is valid for all the post-translational modifications. However, unfortunately, not all the requests come to our desk, so we have no control if the regulators decide that this substance deserves to be called this way, amoxicillin. We never judge if it is an amoxicillin. It will be the regulators in Chile who decide, okay, this is an amoxicillin and can use the same name. Don't be afraid, because then we go to the step further. We have done some work. However, it has been said here that naming of biosimilars, it's a responsibility of WHO INN program. And indeed, the WHO INN program exists to facilitate the identification of medicines so that they have a unique name that is globally recognized and that they are prescribed and used rationally. At present, SBP is the fantastic definition given by WHO, similar biotherapeutic products. We will always like to complicate. Well, at present, SBP are not specifically named, but general naming principles are applied. We realized over the last 5 years, I think, that it was not sufficient, because we have been approached by many stakeholders, regulators, and many people requested us to work in a certain direction. So we have done some thinking, and we have decided that the current policy, that in practice means that some SBP have all that are unglycosylated have the same proprietary names as the same non-proprietary name as the reference substance. The fact that glycosylated SBP are likely to have different name from the substance of reference, and that different interpretation of these policies have led different countries having different non-proprietary name from the same products. And the fact that when different non-proprietary names are used, the reference substance to which the SBP is compared is not identified, Well, this has somehow an impact on the INN program. We cannot just say this is not our responsibility, because how can we know that this epoetin lambda was referenced to epoetin alpha? Now we know because it's one, but in future we will have so many. How we will recognize even what is the reference substance? This is what is happening now. It's just summarizing what I said, and that if we have a generics, I mean chemical, we have the same INN as the reference. If we have filgrastim, so just what we call SB type 1, we have the same INN. It's a simple amino acid sequence. It is always true, but not always in reality, because recently the United States have approved TBO filgrastim. So to increase the complication, they have decided to add two prefixes, at least I know two, one to aflibercept, and the other one to filgrastim, simple amino acid sequence, but they have decided that they deserve a prefix. They were not even glycosylated, but they have decided to do so and to increase the complication. If we go to SBP type 2, well, they are comparable. They are somehow have the same, well, they have the same amino acid sequence, but they have differences in post-translational modifications, so they have the same INN with the Greek letter. Non-SBP, what we have discussed so far. So they are not comparable. They have amino acid sequence that has not been compared, similar to different. So for us, it's a different INN. Problem is that nobody has ever come to us to say we have this. And then, okay, without discussing about the bio-questionable. So it's a very confused situation. It's a very confused situation, and I don't deny this. There is a need to distinguish SBP, and is it an INN issue? Well, how can we distinguish an SBP from each other? They have the same substance name, you have said. For you, it's the same substance. And you are right. And you also mentioned the fact that, of course, in Europe it is said you should prescribe by brand name, but it's not always the case. And as prescription by non-proprietary name is common, and in some countries is even encouraged, this issue indeed does affect the INN program, because how can you trace it? How can you recognize? How can we comply with our mandate? And then if individual regulatory bodies, which is what is happening now, develop their own naming policy for SBP, there will be a diversity in the names, not a unique name which is globally recognized. It is already happening. We have BS in Japan to identify the biosimilars. We have SIM in Australia, and we have other countries that are very creative, like the US, adding prefixes not only to somehow biosimilars, but they don't name biosimilar. So far, they have said to me many times, we don't name biosimilar. So we have just done something for safety, or I don't know for what reasons. And they, to complicate, even added a prefix to an innovator substance. So we have now trastuzumab emtansine in Europe, and adotrastuzumab emtansine, the very same substance in the US. Fantastic. Just see this. I have taken this from Roche, but I think it's fantastic because it shows for the epoetin alpha that we have in Europe two epoetin alpha biosimilar, one epoetin zeta, which is called zeta, but probably is an alpha. And then we have an kappa, which is epoetin alpha biosimilar 1 in Japan, but is called as an INN kappa. And then it has a name for biosimilar. And this epoetin alpha is called epoetin lambda in Australia. Is it a unique name? To me, it is not at all. And so we have completely failed. We didn't maintain, we didn't achieve what it is in our mandate. Because of that, although it is difficult to change positions in WHO, believe me, I am a very pragmatic person. I have 4 children. So for me, there is a need, we have to find a solution, and we have to find it quickly. It doesn't work like that in WHO. And perhaps it is correct that it doesn't work like that because we have to create consensus, we cannot impose everything, but sometimes it's, okay, and I don't say more than, so you were complaining about your situation in the region, but imagine the situation in the global world, it's even more, and I don't know, but okay, we have done some work, we have done some people supporting us, we have done this with regulators, and finally, it is under discussions, a proposal, and I am very diplomatic, that all SBP should be clearly identified. All SBP should have the INN as of the reference product as first part of the name that effectively and unambiguously identify the reference product for this SBP. And then a parallel system could be developed, creating a biological qualifier. BQ is my invention because there was BS, SIM, so I propose BQ, which is more neutral. I don't know if it will be accepted, but this is the way we are calling it now. A biological qualifier that could uniquely identify the substance. And maybe I appear very demanding, but we have been asked, and I think this would be the good direction to go, to apply this for all biologicals. Because even those countries that don't call what they are approving biosimilar, but in reality they should use or could use the same BQ for the filgrastim, for example. And of course, there was discussion in the group about how we should implement this system, and the most two viable options are that the biological qualifier is assigned by WHO INN program according to an agreed policy. Another proposal was also that the INN programs produce an advice document laying out an agreed naming system which may be applied by regulatory authorities. I think that the first proposal was considered more more better, we don't say better, because easier, I don't know, the regulator said it is better if this is in the mandate of WHO if you develop this, so we have quite some work now. And the conclusion, and I think this is the last sentence, is maybe the conclusion some people are expecting because you have heard this presentation before, is that this is an issue that does need to be addressed globally and soon, while the number of registered SBP is relatively small. Not to improve or to contribute to, again, a poietin beta call, whatever, following the alpha example or other examples. That the INN program, WHO INN program, has a clear mandate. And finally, after some time of discussion, the INN expert group agreed to this principle and recommended WHO Secretariat, this is a very diplomatic WHO style sentence, to further work on principles of establishing a biological qualifier system in conjunction with exploring its feasibility. Personally, because I am a mother of 4 children, I have already done all the necessary to do so. We have already the database in place, we have already worked on many, many things. But I cannot go further. I just wanted to show my management that it is possible, that we can handle, that it doesn't require additional resources, that we have to work very closely with regulators on this, and that we can do. So the system is in place, my ideas are in place, with maybe, of course, this is the ideas that we have so far and can be improved. But it is already a lot. In this sentence that you read, you don't see the amount of work that there is behind, that the expert accepted that there is a need of a system and that we have to prepare a draft, explore the feasibility. It means we are working on it. And this would entail, certainly, further consultations with the major stakeholders, including but not limited to regulatory parties and industry, of course. And well, we have quite some challenges, and I'm finished. Certainly, the complexity of the substances, the number-induced difficulty, which means we have 300 maps on the market and we don't know how to name the next one that comes. The emerging of new types of substances that deserve new policies, like for the cell therapy, and we are working on it. And then, of course, this biological qualifier, which is now the one preferred versus the international set of standards, and we are also working on it. I think that, if I may say so, I really need the support of all the people who are willing to go in this direction, because I personally have my ideas, but it's not all WHO, and we have to build a consensus. And to make everybody understand that this would not punish good companies. It would not punish biosimilar producer. It would not prevent prices from going down. It would just protect patient safety. It would just, let's say, achieve the mandate of the INN program, ensure clear traceability, clear identification. It's very simple. And well, in my opinion, we should go in this direction. So I hope we will be able to do together with the regulators, because we cannot do it alone. Muchas gracias por escucharme hasta ahora y disculpa para no hablar su idioma.
Bueno, ahora tenemos la sección de preguntas, si tienen alguna consulta que hacer.
Our attorney Amgen. I was a little confused with the rationale for glycosylated versus unglycosylated proteins. For a glycosylated protein, you can make the conclusion that they're highly similar but not identical. And for unglycosylated protein, you can make the same conclusion, that they're highly similar but not identical. You still can't show identity for unglycosylated proteins. So why is there this distinction between different ion names for glycosylated proteins, but not for non-glycosylated proteins?
Of course. First of all, I said in the very beginning that this is an old program. When we started to use these Greek letters, science was not as much developed as now, and we knew very little. The INN expert wanted to take a wise advice, a wise or precautionary way of thinking, and they said, oh, we have to prevent, certainly glycosylation has an impact, so we have a different Greek letters. But then there were more chemicals people than biologicals at the time, and they said, okay, we name this is this substance, this amino acid sequence, full stop, knowing that we name ideal substances, not the product, not the reality. And I think that was their thinking. Okay, the amino acid sequence is there, it can be reproduced, we don't care of impurities, and that's it. And this is how the system developed. It was considered good until we had one filgrastim. But although in the United States they never approved biosimilars, they have on the market a biosimilar filgrastim or a copy or whatever you want to call, and they have decided that the difference should be highlighted in the name, adding TBO filgrastim. I don't know if they realized that they were naming Teva Biological Organization filgrastim. Perhaps they didn't. I hope they didn't. But this is what they did. So I'm not defending the system as it is now, but this is as it is. And I think the logic was that one, many years before me. Did I answer your question?
Not quite, because it's where do we go forward with this?
I think that, well, as you see in my proposal for the biological qualifier, the discussion which is currently happening is that we should have it for each biological and not only for biosimilar. And I think that this has been said by some regulators when they raise, okay, you're solving a problem, but you're not solving all the problems. And even by generic producers, they said, if you would be using this BQ, or whatever you want to call this, BQ is my invention. It doesn't mean that this is what we are going to adopt. But if you want to use this BQ for biosimilars, you should use for all biologicals, and we wouldn't oppose to it. Which I think it's a good, it's the first, and is EGA, not the American Association, but EGA came to this conclusion that they wouldn't oppose, and I said, okay, and in a way, they are in the same line as IFPMA. And there is still some work perhaps to do in the US. You were going back to the US. I think that there is not a clear understanding of what we are trying to do, also because journalists, Bloomberg or whoever, they are bombarding with invention, the senator, citizen, whatever. So this, I think it should stay out. And that is something that it's irritating me, that I am becoming more important than what I am, and the INN program is taking more responsibility than what it should do. We just want to do something very simple to protect safety of patients and to ensure that if you are taking this and then you are traveling somewhere else, you can have the same. That's it.
Señorita Baloco, primero que nada, congratulaciones por su presentación. Mi comentario es de una tremenda confusión que tengo, porque usted habla de nombres, non-proprietary names, y toda su nomenclatura que da como solución para nominar los productos biotecnológicos y los biosimilares son en base a eso. Usted deja fuera los nombres de marca. No son su problema, no son su preocupación, no los acepta el sistema. Eso me pareció entender. Sin embargo, en el mismo sistema en que usted está incluida, en la mañana la señorita Solenbrink, que hizo una brillante presentación, nos explicó la recepción que ellos hacen de los productos de la la farmacovigilancia, donde reciben productos de marca y los procesan y los reconocen como tales, productos biotecnológicos. Entonces, al parecer, es necesario un esfuerzo de coordinación entre lo que usted representa como INN y lo que representa la señorita Solenbrink como la recepción de la farmacovigilancia.
No sé si es un comentario o una pregunta, pero quiero comentar a su comentario que he dicho que la DCI no es el único indicador de farmacovigilancia, por nada. Yo pienso que debería ser hasta el nombre marca, el batch number. Hay muchos indicadores que son importantes. Yo no soy una experta de farmacovigilancia, pero el problema es que los sistemas no están desarrollados de la misma manera en todos los lugares. Se han tenido 49 muertos. Y no sé cómo decirlo más gentilmente, porque fatalities is in English, pero muertos son, en Tailandia, para utilizar una epoetina alfa. Y el problema es que el sistema de farmacovigilancia ya es tan, o era tan feble, no fuerte, no sé cómo se dice, débil, que no han podido saber si era en la molécula originaria, si era un problema de producción, si era un biosimilar o cualquier era el problema. Eso yo no estoy defendiendo la utilización de INN contra la utilización del nombre de marca. Bien sûr, mi trabajo es hacer denominación común internacionales. Y hay un problema en la utilización del sistema en el campo de biológicos que se muestra evidente ahora. Entonces, ¿cómo solucionar este pequeño problema? Sin decir que en la farmacovigilancia uno no tiene que también tener el nombre de marca o el brand name. No he dicho esto, lo siento si el mensaje que transmití ha sido esto. Al mismo tiempo, los comentarios negativos que hemos tenido es que nosotros no damos denominaciones común internacional, es decir, non-proprietary name, que no pertenecen a ninguna persona. Y dando el BQ, nosotros decimos que eso es eso. Pero la sustancia va a ser epoetin y después va a ser BQ de la manera que desarrollamos el sistema. B, no sé cómo va a ser, o por lo menos hemos discutido un poco, pero no tenemos una clara visión hasta ahora. Con eso queremos simplemente identificar qué es qué cosa y permitir a que todos lo hagan. ¿Cómo lo pensamos de hacer? Y eso es verdaderamente solo lo que hemos discutido, pero no significa que es lo que hemos decidido. Hemos pensado de publicar eso como un anexo al listado, pero el anexo no va a ser muy útil. Entonces, desarrollar una base de datos en colaboración con las autoridades regulatorias que quieren darnos la información. Por ejemplo, esa epoetina que tiene su BQ, ha sido aprobada en eso país. Y recordando que nosotros nombramos las sustancias, no los productos. No sé si puedo responder, es que es muy complejo, estoy de acuerdo, y no sé si tenemos la solución ideal, pero tratamos de encontrar una solución, por lo menos.
Muy cortito, sí. En su presentación final, en las conclusiones, no aparecen los brand names, figuras, fíjense bien.
La última frase.
La última conclusión.
Ojo, esa conclusión es lo mejor que puedo decir para ser muy diplomática. Y no quiero explicarle por qué tengo que ser muy diplomática, pero que es que hasta en la OMS no todos están de acuerdo que tenemos que ir en esa dirección. Entonces, yo tengo dos niveles de supervisión. El nivel más alto está de acuerdo, el nivel de medio, que es un virólogo y que nunca trabajó en medicamentos, no entiende y está asustado de esa idea. Entonces, eso es lo mejor que puedo decir por el momento.
Karin Heidenreich from Novartis. I fully appreciate your hard work and you have my full sympathy, but my comment goes actually in a similar direction. I don't believe that unique identifiers, BQs or however you want to call them, will really bring a globally harmonized solution. Solution for what is the question? I don't believe that, let's say, the US, Japan, Australia, UK will change their systems they have already in place. So I believe it will even create more complexity with a new system, with a new identifier that is not transposed by everybody. And we heard this morning that Brazil is not following WHO INN naming at all, and there are certainly more countries like that. So for pharmacovigilance, as we said also several times, that the brand name, that batch numbers, that other identifiers are absolutely needed to really have a clear identification. And what I believe a good solution would be to achieve that is actually proper training of healthcare professionals and of authorities to really make it happen. I think for me this is the key issue. If we now introduce a new system, again you need training for all these people to adhere to the system. Why not to use systems we have already in place and really make them stronger?
I have to say that we have not done this alone. So, Japanese will go in our direction, sorry. Australian will go with us. Other authorities that have been, because we have done together with them. We had a meeting with them and we have discussed how can we sort it out. And some of the authorities I cannot deny, and I can say the Koreans are very much against. Some, like Japanese and Australians, they are those that have a system in place. They said, if you go in this direction, we will follow you. Some Europeans, they are somehow revisiting their thinking, and when we asked them, would you be willing to go in this direction, they replied, I cite Peter Richardson, it is unlikely that we are going to use, but we cannot say no. So there is some building of consensus, and this is why it's taking so much time. And just because I know what is your company, so maybe it's somehow related to the apoietin alpha lambda problem. That problem, the Australians told us, would have been never appearing if we would have this BQ system in place. So I am not trying to invent something for the pleasure of doing, really. For us, it's more work. It has been a nightmare to receive letters from all the world, and in this, and it's not even internally, it's not always pleasant. I would be happy to remain on my INN request and do my job and go home, but I think there is a need, and I think that WHO has a responsibility. And by the way, just to explain you how we think, that we will not impose anything on anybody, because either it is by consensus or it doesn't work. So we are drafting now something that we will present to a broad group of regulators on the 7th of April for finalization, and then to the experts. And once this document is final, it will not be published as a decision. It is going out for global consultation. So there will be a kind of the word that somebody in WHO likes very much, dissemination plan for having comments from everybody. So, and I don't see that by April we will have decide, because then we will receive comments. And the idea is not to create another system, but is to avoid all the different systems existing, and they exist. And I don't think that just improving pharmacovigilance we will solve everything, with all the respect for pharmacovigilance. And not because I don't want the brand name or the trade name in the pharmacovigilance, but in some countries we have to be realistic, and I think that the pharmacovigilance people, they can say that they only report by INN. It will take time until this will change. And the 49 fatalities in Thailand that are a striking example, in my opinion. I think nobody could trace back what was the problem, because it was a poietin alpha, full stop.
¿Otra pregunta? ¿Alguna otra pregunta?
Muy bien, bueno, pues muchísimas gracias otra vez por la discusión tan interesante y apasionada también en algunos momentos. Gracias, doctora Baloco. Y bueno, yo lo que sí quiero es mantener el tiempo, así que les voy a dar 15 minutos para el café con el objetivo de acabar a la hora. Muchas gracias.